If you'll look at the agenda, and we'll be following this agenda, no surprises here, we're kicking off with Dr. Bryan Kolb. So Dr. Kolb, will you come forward? Just the little bit that is there is Dr. Kolb is from the University of Lethbridge. He's on the Board of Governors and the research chair. And I'm very curious, and maybe you'll tell us before you come down off the podium, what it means to be an officer of the Order of Canada.
Thank you. Good morning. The Order of Canada is the highest civilian honor you can receive in Canada, so that's what it is. And this little pin is we're supposed to wear at formal things, and it tells Mary Lou and Marlene, wherever she's sitting, the only ones who would know what that actually means. But that's okay. I'm following the rules and wearing it. It's a pleasure to be here. And before I start, I should give you a little background, and that is I gave a talk to grand rounds nationally last December to neonatologists at Calgary Children's Hospital, and it was broadcast across the country, and on plasticity and various treatments you could use and so on. And I was stunned to discover from the neonatologists that the standard of care for kids who have hypoxic ischemic events at birth is cooling for 24 hours and send them home. And I said, this isn't very good. We need to have post-hydrotherapy plan. And so we're now working, trying to set up a program for these babies that will begin the moment they're warmed up again and continue on. And Mary Lou will get this. We applied for money. We were told you can't get money to do this because you have no preliminary data. How can we get preliminary? And you can see the problem. But it's not just the epilepsy kids. A lot of these kids will develop epilepsy, of course, but that's not really the issue. Okay. So one of the things I learned, I took my first course on the brain in 1966, quite a while ago, and one of the things we learned is that your brain doesn't change. It's, you got a brain, and that's what you've got, and get used to it. And of course, over the years, we've discovered that this is not true. The brain is constantly changing, and I tell my students the reason that at my age in my 70s that I hope you don't see me as demented is because the brain is changing. Otherwise, things wouldn't be going so well for me. And they sort of look at me like, what are you talking about? But the idea here is the brain is constantly changing, and it has to change. So, whoops. I don't know what I did to get that, but anyway, the idea here is that if you learn anything, the brain has to change. So whether you're learning someone's name, learning some sort of motor skills, learning to play, I didn't do that. Golf, learning anything today, the brain must change. So in order to learn something, there has to be a structural change in neurons in the brain. It's not me? Okay, this is an example of how the brain changes. So if we look, this is studies done by Ernie Scheibel at UCLA some time ago. Basically, he was looking at post-mortem tissue in humans. And one of the things, you can see that there are I think the little one, yeah, there are neurons here that are cartoons. They're more complicated than that. But this is really simple, this is more complex, and this is quite complex relative to the other two. So the idea is that the complexity of neurons in the brain is related to the complexity of the computations that those neurons are engaged in. And, okay. Okay, it was so good the first time, we'll do it again. Okay, thanks. And so the idea is that the more complex the computations are, the more complex those cells have to be. Now, we just heard that kids with various types of surgeries to reduce seizures have language problems. And so why might that be? Well, if we look at the effect, this is the language area, and you can see that the cell here is more complicated than the cell in the finger area, controlling my finger use, or in the body, the trunk area. And that makes sense. What I can do with my trunk is nothing like what I can do with my finger or I can do with respect to language. So if we look at this neuron, it's complicated. So we look at the effect of education in their studies. the more education you have, the more complicated these neurons in Wernicke's area are. And that makes sense. And not only that, if we look at a sex effect, one of the biggest effects that you can see at sex effects in neuropsychology is related to language. So little girls start speaking before little boys, and I can be sexist and say, and never stop. And one of the reasons is because these cells are much larger in females than in males, okay? And we can also look at occupational effects. So what they did was to look at people who were stenographers, and for those of you who are too young to know what that is, it's people who made their living typing, okay? So they're expert typists. All of us type now because we have computers, but the woman who used to type our stuff could type at 125 words a minute and never make an error, okay? So that's pretty good finger use. And they compared the finger area in those ladies to other ladies who were sales clerks. Similar education, similar intelligence, but the size of these neurons in the finger area were about double in the stenographers. So you can see that experience is changing the structure of these cells. These ladies did not have big cells, presumably, before they started typing. Okay, so why does this happen? One of the things that experience does is it increases brain activity. So if you're doing things, the brain is active. Second thing is that brain activity changes gene expression. What does that mean? If you think about it, if you remember any biology, all of the cells in your body have the same DNA. So the cells that made my skin, my hair, my eyes, my brain, my bones, all have the same DNA, but the cells are different. And the reason the cells are different is that different genes are turned on or suppressed. Okay, so that's gene expression. Experiences change gene expression. So if I'm successful this morning, I'm getting into your cells and changing the expression of DNA in those cells. That's important because we haven't changed the DNA, we've simply altered what's actually active. And experience alters gene expression directly, so it can actually work directly or it can work through the increase in brain activity, and that gene expression changes the structure of cells. So this is a cartoon. This is a neuron. The cell body isn't that big relative to the cell, but just pretend it is. These are dendrites, so they're the receptors of the neuron. So there's an input from some other cell. So imagine you're getting visual input. It goes from my eyes to, let's say, the thalamus in the brain, the thalamus to the cortex. So we're looking at a cortical neuron. It's getting input from, don't do that, from other places in the brain. And in addition, we have a direct effect of the experiences on the cell body. That changes gene expression, and that alters the structure of the neuron. Okay, and you're going to see how this works. So there are some rules in brain plasticity. So I started studying brain plasticity in about 1986, 1987. And what we were looking for is rules that would guide our understanding of how the brain changes. And I'm going to give you some of those rules. Hopefully I can make them simple. So behavioral change reflects brain change. If behavior is going to change, the brain must change. But there's a corollary of that, and that is if the brain changes, behavior changes. Okay, the two go back and forth. And so the key point here is, or a key point, is that you can have a behavioral change that's caused by more than one, the same behavioral change by more than one plastic change in the brain. That's great, because it means there's more than one way to skin a cat here. Depending on the details of an injury you might have or the surgery you had, the changes in the brain may be different. What does that imply? It implies the treatment must be different too. You're trying to treat a particular kind of brain tissue. So, second is all nervous systems are plastic. Even tiny little worms that have only 302 neurons, these worms have plastic changes. So what does this mean? It means that we can actually study non-human models and learn how the brain is changed and how we can control those changes. That's an important point. I don't study worms, but I do study rodents. Plastic changes are age-specific. This is important too. So the brain is changing from the moment that it's conceived, the moment the brain starts to be produced. And the changes in the brain are going to be different depending on the age of the brain. So the changes that are occurring in my brain are different than the changes occurring in my brain when I was 6. And the reason for that, of course, is that the brain development, we used to think that brain development was done by, 15, 16, maybe 18. We now know that it goes on at least until age 30, and according to my wife, in men, probably closer to age 50. I guess I'm done because I'm long past that. But the idea here is these changes in development go on a long time, and they're age dependent. So making a brain is a miracle. It's got a lot of neurons, 86 billion neurons, and approximately 10 to the 14th connections. That's a lot of connections. You could not have a blueprint for something like this. What you have to have is something that can modify itself. So what the strategy that Mother Nature came up with was, let's make something that's twice as big as we need. This is totally different than making a heart or a bone or anything else. We make a brain that's twice as large as we need and get rid of half of it. Why do we get rid of half of it? Because when you're born, the brain doesn't know whether you're going to be living on the North Pole or the equator, for example, and the requirements are not the same. So we're going to see that experiences are going to sculpt the brain. Some of those experiences are hormonal, some of them are external, and so on. There are a whole series of events that have to occur. You can read them. You've got to make the cells. They're born in the nursery in the brain, which is the subventricular zone, and then they have to migrate to where they're going to be. I'm going to be a visual cell, and they migrate to where they're supposed to be. I'm going to be a motor cell, and they migrate there. We have to mature. They have to produce synapses. But the really interesting thing is this, and that is getting rid of stuff. I think I don't have that slide. Okay, doesn't matter. If you look at the pruning, the getting rid of neurons and the getting rid of connections, we have an increase in the number of connections up until about between age 2 and 5, depending on what part of the brain we're looking at, and then we start getting rid of them. Now, we get rid of them at an amazing rate. In the prefrontal cortex, this stuff up here, prefrontal is a stupid word. What would it mean? In front of front. But that's the word we're stuck with. It's the stuff up here. It's at the very front. The prefrontal cells start pruning early, maybe 5 or 6 years, but they really start with adolescence. My least favorite group of people, and I've told my daughter this, are 12-year-old girls. And those of you who've had 12-year-old girls know why. I had 2 sisters and they were younger, and I knew why earlier. There's a lot of changes going on, and during that period, these kids are losing approximately 100,000 connections per second. 100,000 cells, 100,000 connections, 100,000 connections. And I can recall saying to my wife, going to bed, why did we get involved in this? And then the next morning, there's a different person having breakfast, and you go, who's this person? That's not the person that was having these temper tantrums because we're all idiots last night. So these changes are occurring. In boys, it's a little later, and it's not quite as so severe because they don't have this huge change in hormones that we see in girls. But that's the point. You're pruning, and this pruning continues until probably age 30. It goes on for a long time, and if I ask people who are over 30, ideally over 40, when did you become who you are? People will say, women will say my late 20s, men will say my early 30s. And for me, I would say it was 32 or so is the person I became. So when I first started professing as a professor, I was 29 and I was not there yet. I can tell you, looking back, I can't believe students paid to listen to me, but they did. Okay, so prenatal and preconceptual experiences count. So things that happen before you're born alter how your brain develops. Not only that, the recent studies we've been doing are showing that things that happen before you're conceived. What the hell does that mean? It means your parents. And it doesn't mean your mother. The biggest impact on your brain development from preconceptual experiences is your dad, believe it or not. And this actually crosses generations. So the experiences that your dad had, good or bad, before you were conceived will affect your children. This is a very interesting phenomenon, but it means the nature of recovery post-injury is going to be affected by things that happened that you had no control over, that happened before you were conceived. Plastic changes are brain region specific, so the changes that occur vary depending on the actual place in the brain that we're looking. experience-dependent changes interact. So we call this metaplasticity. Here's the deal, and it's one of the problems I have as a neuroscientist working with lab animals as well as people, but with the lab animals is we tend to give them one experience. I'm going to give you crack cocaine, and we're going to see what happens in your brain. And then I'm going to do another study, and I'll give animals tactile stimulation. I'll talk about that in a second to see what happens in the brain. That's not how human life is. We have one experience, and then another one, and another one, and another one. So experiences are being affected by things that happened earlier. That's called metaplasticity. We're really ignorant about the details of this, but it's really important for the kids we're talking about today. Plasticity is related to the intensity of experience. So if you're learning to play a musical instrument, you can practice scales once, and then what? You don't know them. You've got to do it over and over and over and over again. So people are expecting miracles. People will say to me, my mom had a stroke and she doesn't talk, and when's that going to happen? I say, well, how long has it been? 6 months. How long did it take her to learn to talk the first time? Quite a lot longer. So the intensity of the post-injury treatment or experiences makes a huge difference, and we have to keep that in mind. We can't worry about the fact that things aren't changing quickly. And plasticity isn't always good, and I'll give you some examples of maladaptive plasticity. Well, one of them you're familiar with, and that is the delayed onset epilepsy following brain injury. You have a brain injury, and 10 years later you start showing seizures. There's something percolating in there that's not good. Okay, why can I use lab animals to study brain plasticity? Well, one reason is I told you that all brains are plastic. And so we can do a lot of things. We can actually, this is a rat who's learning how to reach through a slot here. I'll say a little more about that in a second. These are neurons. They're gorgeous things. And we can actually look at the structure of these neurons and make all sorts of inferences about what they're doing. Remember, I told you complexity of the computations is related to the complexity of the neurons. And then we can look at epigenetics, that is changes in gene expression, all in the same animals. So if we look at good things, one of the remarkable things, and in my experience, the biggest impact on brain is tactile stimulation. So we look here at various forms of tactile stimulation, which we can call Kangaroo Care, for want of a better word, for an obvious reason. So we can have massage. So we're massaging the mom before the baby's born. I'll explain why this would work. We can have skin-to-skin contact, which is one of the things that we're working with the NICU neonatologist to make sure there's skin-to-skin contact as soon as these kids are warmed up. And this goes on every day for months. But in rats, we can either, this looks a little nasty. It's an artist's rendition of a child's hairbrush. It's not spikes. So we can do the same thing that's going on here with this mom. Here's a baby that's being massaged, or with a baby rat, and we can use a little brush. Now, why would touching the skin have any effect on the brain? The skin and the brain developmentally come from the same germ cell there. Okay, well, so what? Well, it means that they're going to respond to the same things. So if you fall and scrape your hand, what do? You rub your hand. Why do you rub your hand? It feels better. Why does it feel better? The skin is producing various compounds, but my favorite one is called FGF2, fibroblast growth factor 2, and it starts reparative processes. What's that got to do with the brain? Remember, skin and brain came from the same place. The brain responds to FGF2 as well. So when you start stroking your skin, you're making FGF2, it's going in there. So what we've been able to show this is that there's a direct connection between skin and brain. In fact, you don't even have to touch the skin. You can use a wide spectrum light that's used to treat burns, and that actually will affect the brain as well. So here's an example. Tactile stimulation, even preconceptually. So your parents are given tactile stimulation, affects how your brain develops and how your children's brain develop. So here's an example. Here's a rat that's learning to reach for food, or it's learned. So it reaches through the bars to get this chicken feed. And because it's like picking up tennis balls or golf balls in a big barrel, and you're only allowed to reach once, you're not going to just take one. People will try and take more than one. And so they're going to drop them. Rats have very good dexterous digit movement. So they're, in this kind of situation, they're at about 50%. These are control animals. These are their litter mates who had 2 weeks of tactile stimulation, 15 minutes, 3 times a day, postnatally, and then nothing happened for 6 months, and now they're being trained to do this, and you can see they're way better. Okay, this is an effect that's chronic, never goes away from this early treatment. Okay, well, what if you had early brain damage? Would, in fact, this make a difference? And the answer is yes. So here are the control animals. Here, about the same place as before. Here's the animals who got the tactile stimulation. Here's the effect of the brain injury in these animals. These are prefrontal injuries, and so they're really terrible. They're at about 25% accuracy. But if we give them 2 weeks of tactile stimulation, just like we did in the previous slide, and then come back and look at them several months later, you can see that they're as good as the control animals. That's a remarkable effect. The brain has been changed, the structure of neurons has been changed, we can see changes in gene expression and so on. So this tactile stimulation is a powerful thing, and it can be initiated immediately in these children, or in this case, these rats. Okay, language and executive function training. Language training is really important. We heard about that already, but one of the key parts to this is what I'm going to call serve and return. You have to talk to the child, so you're going to be the child. And I'm going to talk to you, and I'm going to say, what do you want to do? And whether or not the child can understand, it's back and forth. There's eye contact, there's often touch, and so there's tactile stimulation. It's not just language going on around the kids, has to be directed at them, and that's a serve and return kind of thing. In addition, we have what I'm going to call executive training, and that is training and planning, training in a variety of little task games, peekaboo games, and so on, that make a huge difference. And we have a program in Lethbridge that my colleague Robin Gibb is engaged in. The whole city's now involved in it, training 3-year-old kids on a variety of executive tasks. Are you going to talk about this? No, you're not. Okay. Executive tests. Early on, you train the parents, and now it's a game that we have a serve and return. The kids are playing this game with their parents, and Robin's been able to show that after a year of this in school, these kids are way ahead in terms of school readiness. So the same logic here for kids with brain injuries. We can begin these executive training at age 3 and a huge impact. But not everything is good. So bad. Psychoactive drugs. So years ago, Terry Robinson from the University of Michigan gave a talk. He's a close friend of mine on the cause of addiction is changes in brain chemistry. So I listened to this politely, and then we went for a walk afterwards, and I said, Terry, that's the biggest pile of crap I ever heard. Addiction is not chemistry. Addiction is learning. There's a morphological change in the brain that causes addiction. He says, well, no one's ever showed that. So I said, send brains. So he did, and luckily I wouldn't be telling you this if I wasn't right. I was right. Every psychoactive drug we've looked at produces a change in the brain that's permanent. Caffeine has a really trivial one as an addict to caffeine. I'm saying it's really trivial. But it's not so trivial with nicotine, and of course nicotine is very difficult drug to get off of. So we've looked at lots of drugs. Here's an example of amphetamine. So if we look here, this is prefrontal in the rat. This is a cell. I hope visually you can see it's more complicated. In an animal that got amphetamine for 2 weeks, and then we looked 4 months later at the brain. And there's a permanent structural change. This is an animal that got saline. So we got all these, and you're going to say, wow, you said complexity was a good thing. Yeah, but I also said that different parts of the brain change differently. If we look at a different part of the prefrontal cortex, the stuff behind your eyes, it's the reverse effect. The cells get simpler. So what we're seeing here is that these psychoactive drugs are remodeling the brain. And this has an effect on later plasticity. So what kinds of drugs have we looked at? You name it, we've looked at it. All of these drugs produce changes. They're not all the same changes. The same class of drugs produces a similar effect. So stimulants have an effect. So cocaine, nicotine, amphetamine have similar effects, but they're different from, say, opiates, morphine, for example. Students worry about this and they say, oh no, I've been smoking pot. Is this really bad? I'm going to say it's a little late to start discussing whether it's bad. We just need to know that it's changed your brain. And It's really bad if you started doing it at age 13 or 14, which in Canada right now, since we've legalized cannabis, is a big worry, that the brain is changing during a time that it's already producing changes because of pruning. You don't want to be adding drugs at that particular moment at all. Anyway, stress. Stress is a big one. And for those of us from Canada, the biggest natural disaster until the Fort McMurray fire a couple of years ago was the Quebec ice storm of 1998, I think it was. Yeah, 1998. So you can see what happened. We had power failure that lasted about 6 weeks in a record-breaking winter where the average temperature was about minus 20 Celsius, which is cold in Fahrenheit too. I'll just leave it at that. And so there was a problem here. Many pregnant women were at various stages of their pregnancy. They may have been early, first trimester, second trimester, third trimester, when they were really stressed during this period. Some had no stress because they went to Toronto and stayed with relatives until the power was back on again. Other ones didn't have that option. So what this group at McGill did was to simply look at the outcomes from these kids. And what happens here is that, here's the Quebec ice storm. You can see what the problem was. It just totally destroyed the hydro system, and that's why it took so long to repair. What they did was to follow the infants who were measured at 2, 5 point, you can see it, various times, and now they're looking at them 21 years later to see how their brain is different. The bottom line here is behaviorally, these kids are very different, especially the ones who were from the second trimester, where the brain is really developing. The kids who were in the first trimester, not so much. And see, what you can do is you can have a mom who had children before the ice storm and compare the two children, the ones who were gestational during the ice storm and the ones who were not. And there's a huge effect on behavior, and there's a huge effect on a range of behaviors, huge effect on gene expression, and they're currently analyzing the structure of brains from MRIs. These are soldiers, US soldiers, who have PTSD coming back from Iraq and Afghanistan. The key point here is the places that have arrows. Blue means the brain, the cortex is really thin relative to control. So if you look at this region in prefrontal cortex, in a veteran who has PTSD, it's really thin compared to one who didn't, and it's really thick. This is not permanent, it's changeable, but the point is that that experience has changed gene expression and has changed brain structure. Learn non-use. This is really important for our kids. If you don't use it, you're going to lose it. Here's a nice experiment by Randy Nudo. Here's a squirrel monkey brain, and what they did was they made a small injury in the region of the brain controlling digits, okay? And then some of the monkeys got therapy, and some of them did not. The ones who were forced to use the injured, the limb contralateral to the injured hemisphere, the treatment, this is the size of the motor control, it's maintained, and the hand and digit area actually increases. These are the monkeys that had no treatment. It's actually gotten smaller, so we're going to call this learn non-use. You don't use it, the brain starts to retract, and the effect gets bigger and bigger, which is one of the ways to solve this is constraint-induced therapy. And constraints. This should show us, yeah, this is OJ. We can see the movie. This is OJ was hit by a car, and it used to be found. I'll tell you what happens to OJ. OJ, I'm going to act it out for you since we don't have the movie. So OJ, after the injury in that movie, is walking like this. Okay? 6 months later, here, oops, didn't want to do that. 6 months here, OJ runs down the stairs and runs outside, and the only way you can tell that OJ is not normal is by looking to see if he has 4 legs. And no, he only has 3. So what's happened here? We have compensation, and that's what we're going to see after brain injury is compensation, not recovery. Recovery doesn't really occur. It's compensation. So what we have to do is to enhance the compensation. OJ always has 3 legs after that surgery, but he acts as though he has 4. Okay, so what's the likelihood of recovery? Well, this is a cartoon. This is area A in the brain. Here's area B. So there's processing going on here, processing somewhere else, and behavior comes out. If you have a small injury, you can alter the processing here, which will alter the processing in the next region and alter behavior. But notice you can still produce behavior. If everything's gone, so we've lost an entire hemisphere, now we've got a real challenge, because the input that should be going to here is not coming. And so we have to figure out a way to bypass this and get these inputs to go there. Okay, so it's a different problem depending on the size of that injury. So what treatments work? Lots of treatments can work. Tactile stimulation, I've told you, can work. Can it work if I've got a 12-year-old who had early injury? I don't know. We do know that it works in adults who've had early injury, adult rats. We know that it works in stroke patients who've had adult strokes, so likely it will. Constraint-induced therapy, forcing people to use the injured, side of the body that's not functional. You can do this in a variety of ways. And one of the things Monica and I were talking about earlier is that you don't want to start it the day after the surgery. You can actually make things worse. You've got to wait for the constraint-induced therapy. That's not true of tactile stimulation. Treadmill training, obviously. Virtual environments. There's some neat stuff going on at Laval University in Quebec, in which you've got people on treadmills, but you've got this virtual world they have to navigate through, such as going through a park and there's balls coming and there's cyclists and so on, to get them to respond to various things, using robotic devices to move limbs, shaping behaviors, and then the task-oriented physical therapy. All of these can be effective. However, many of the treatments work better in combination with pharmacological therapies. And you're going to say, what does that mean? Well, there's a variety of things that enhance plasticity. Believe it or not, nicotine is one of them. And in combination, that doesn't mean you smoke. I gave a talk for rehabilitation of stroke patients, and somebody put their hand up and they said, "You're telling me massage and a cigarette is the way to treat people after stroke?" And I said, "It's not what I said exactly." And he said, "Well, if I told my wife that if we had more sex, I'd recover faster." I said, "I didn't say that either." But I did say the tactile stimulation, so massage and nicotine in combination work, nicotine patch and so on. This has not been done in children at all. It's being done in adults. It certainly works in rats. So it's one of the things that needs to be tried. Okay, in conclusion, the brain is affected by experience. This is necessary for development and for improvement after any kind of event. We have a wide range of principles that underlie plasticity, and our challenge here is to learn how to apply them to children. We now have a good idea from our animal studies. Now we're trying, and I told you about the NICU program we've begun, and plasticity is influenced by a wide range of positive, stop that, too many green things, positive experiences, but also negative experiences, and I gave you just a flavor of what those might be. Now, we were asked to provide references, and so there are references you can look at that are general, giving a sort of broad view of plastic changes. Thank you for your attention. I hope you learned something and I got into your brain and changed it. Thanks.
Before you leave the microphone, because we're filling our brains and we're going to go right to the panel, Dr. Kolb, while the panel is coming up here and settling, for this crowd, what's one sort of 30-second exercise they can do with their brain to ready it for the next activity? Should they massage the other person?
I'm not going to be responsible for assault, but I would say standing up and moving around and getting the blood moving in your brain is the easiest way to get ready.
Wonderful. So please do that.